The overall goal of the following experiment is to translate Plasmodium proteins using HeLa-based cell-free expression systems and to purify the proteins from microvolumes of translated product. This is achieved by cloning Plasmodium genes to prepare recombinant plasmids for two-step and one-step in vitro translation cell-free expression of malarial recombinant Rhoptry proteins. To translate the recombinant proteins, the prepared plasmid is incubated with transcription mix when using the two-step expression system, which transcribes mRNA. The resulting mRNA is then added to the translation mix for protein translation. While using the one-step expression system, the recombinant plasmid is incubated with HeLa cell lysate for transcription and translation of recombinant proteins. Recombinant proteins are then purified from microvolumes of translation product. The results show successful expression and purification based on Western blotting analysis.
The main advantage of this technique over others, such as the wheat germ expression system and the rabbit reticulocyte lysate system, is that this system can express proteins in 3 hours. No codon optimization is required. It is affordable and easy to use. Multiple antigens can be screened on microarrays, making the detection of antigens for therapy and diagnosis possible.
Generally, individuals new to this method will struggle because it is cell-free expression which expresses proteins in microvolumes, making protein purification extremely difficult. We first had an idea to use this system as we could not express proteins using E. coli and found other